Obviously, for me, it's a great pleasure to be back in Louisville and see my colleagues and friends. And different from when I was here, you actually have a chance to tell you about some of the stuff that we do, because you know how it is when you're sort of home. Today, what I will tell you about relates to work that's been going on in my lab for about really 20 years, where we basically focused on how benzo pyrene, sort of a very close first cousin, I think, of TCDD, has the ability to essentially start processes that give rise to oncogenesis or changes in cell phenotype. And that sort of led to the idea that there's a reprogramming of the genome that takes place as a function of these exposures, and I wanted to understand sort of the molecular basis of that response. I think the talk probably is quite fitting in the conference, because the bulk of what I will talk about really focuses on these three domains within the regulatory network of cells. And I think if you reflect back on sort of the science that we've listened to in the last day and a half, all of it really revolves around things that relate to the biology that's driven through these processes. So in the context of ontogenic differentiation of, in this case, liver, this certainly could apply to any other organ system, we typically think of a differentiation network that's going to give rise to the occurrence of phenotypic features that drive the biology of that organ. Here, of course, when we think of liver, we think of albumin, we think of metabolic functions, and we think of cytochrome P450 metabolism. And these are things that we've heard about repeatedly throughout the course of our visit here. Apoptosis or necrosis, or aponecrosis, obviously is a very important component of the toxic response. We all think about this as sort of the end point of sort of the biology that we're driving at. And then cell cycle, and I think that we've also heard questions about sort of regeneration of liver capacity. What does that have to do with differentiation features? And what we need to recognize is that genetic networks in cells, and as a consequence of that, metabolic function in cells is really the interoperability of cells and the ability of the cells to navigate and transition from one state to the other in fluid ways that allow the cell to carry out its functions. And so the ability of cells to engage, say, in the preservation of differentiation features calls for a cluster of genes to be operational at any one point in time. But when that cell is challenged, as in the case of a toxic exposure, we need to have the ability to transition, genetically speaking, and ultimately functionally speaking, from this differentiated state into a state that allows for the cell to regenerate if it's going to regenerate. And if that capacity to regenerate is overwhelmed, the ability to die. When cells lose the ability to die, we have oncogenesis. And so what really becomes important, I think something that I want you to take with you from the talk, is that cells, the genome of most cells, retain the ability to make these transitions. An easy way that we think of those transitions is epigenetics, for instance. So you change the coding that sits on top of the DNA template, and in so doing, you now have the ability to navigate. In some instances, those transitions call for a mutation that actually changes the codification of protein. And so that sets the stage with the idea that an alteration in the transition of cells across these functional genetic domains is what gives rise to oncogenesis. And I've been sort of interested in studying what the biology is that drives that process. And so we did this using, obviously, a chemical carcinogenesis model. And the molecule of interest for me has always been benzo pyrene. Captured here, this is a polycyclic aromatic hydrocarbon, as I said, a first cousin of TCDD, but with a biology that's probably a lot more complex, relative to even the complexity that you alluded to, Tim, because in addition to the ability of the aryl hydrocarbon receptor to be recognized by this molecule and to actually function in response to benzo pyrene is a very good DNA damaging agent, because many of the cytochrome P450 derived metabolites that originate from here give rise to oncogenesis. And of course, the story that we've all learned when you take a toxicology course, when you think of Miller and Miller, is the formation of the 7,8-diol 9,10 epoxide, which is a very powerful DNA intercalator and has the ability to recognize warnings in DNA, and those covalent adductions give rise to mutations. And we know of some genes that targets. So this adds a tremendous level of complexity to when you think about the fact that in addition to this molecule going through this pathway, it also undergoes formation of what we call benzoate quinones, and those quinones give rise to oxidative stress. So then when you think of the totality of that biological response to a molecule like benzoate pyrene, you actually have to think of three mechanisms whereby BAP could actually give rise to carcinogenesis. It could be through DNA damage, through the conventional 7,8-diol 9,10 epoxide covalent adduction giving rise to mutations, well documented. It could be through disruption of the oxidative balance of cells, and in so doing, of course, maybe moving through a pathway that's along the lines of what Tim just described for you, or it could be through functions of the aryl hydrocarbon receptor. And we've sort of always been sort of bogged down by the complexity, I think, of what that represents. So we established an in vitro model system in which we would take cells in culture, whichever your favorite cell happens to be, and we would do repeated interrupted challenges with benzoate pyrene to give rise to an oncogenic phenotype. And this is a model that's being used repeatedly in many laboratories. So we would do weekly dosings, cells in culture, we do serial passage, and in so doing, we'd be selecting for cells which acquire an oncogenic phenotype. We test for that oncogenic phenotype in vivo, document that that was taking place, and then we go back and track down sort of the genetic changes that are taking place during the course of that transition. And so I was one of the first people to sort of jump into the genomics bandwagon. Many of you know that. And the reason that I did that is because I was always puzzled by the idea that I spent 10 years of my life, thanks to Bob Roth, studying the RAS proto-oncogene. And we can make reference to that, later on, because Bob actually hinted me, for me to go in that direction. So I spent 10 years looking at RAS, but recognize that the RAS proto-oncogene is only one answer to a very complex question, which is really along the lines of what Tim was actually alluding to. And we made one discovery very early on in this genomic adventure that we engaged in that really has dominated what I've done for the next 10 years after that. And that's captured in this slide. What you have here is a northern blot. That's something that probably, remote to some of the younger people in the audience. This is back when you really had to work hard at looking at genes. But we did this experiment that basically focused on genome-wide alterations in gene expression. And what we discovered is that when we did a genome-wide search for genetic targets to benzoate pyrene, this model of oncogenesis, the one alteration that kept repeatedly coming out was the overexpression of repetitive sequence of DNA. And because of the technology that we were using at the time, I actually was biased against it, and I kept thinking this was an artifact, and for 5 years we kept dismissing the piece of data that we were getting, but that's captured here in this northern blot. This is a northern blot for something that we call line 1. This stands for long interspersed nuclear element 1. And this is one of the many repetitive sequences within the genome, and it turns out that BAP and BAP metabolites very readily reactivate this sequence. And now, what makes this really complex is that these are sequences that are actually silent in the differentiated genome of cells. And they're silenced through epigenetic mechanisms. So, for those of you that probably don't know much about transposons, because that's what line 1 is, this is a very large family of genetic elements within the human genome that can be divided into two groups. DNA transposons, which really constitute only about 2 to 3% of the human genome, and then a very large chunk of them, which we call retrotransposons, because these are jumping genes that move using reverse transcriptase and RNA intermediates to actually carry out that jumping function. But notice that this is 42% of the human genome. That means that 50% of what you carry in pretty much every single cell of your body is what we used to call 20 years ago junk DNA. And now we know it's not really junk. And as I showed you from the previous northern blot, it turns out that chemical carcinogens of the BAP family reactivate these sequences in ways that we don't know what that does to the human genome. The one that I study actually belongs to what we call the non-LTR family of retrotransposons, and this is where L1 resides. L1 actually constitutes 17% of the human genome. So of that 42, 17% is line 1. And so here is, I think, something that you probably will need to sort of have a good understanding of so that the data makes sense to you, down the road. Turns out that there is significant conservation in these elements throughout the human genome. The human, compared here to the mouse, we actually started much of this work in the mouse, then migrated to the human for one very important reason. The regulatory region, that region which we're interested in studying as a function of carcinogen exposure, is actually structured in a different way in the human element. And so the bulk of what I'll show you really will be human-based line 1 biology. But suffice it to say that you have a 5' untranslated region in this genetic element that functions a lot like a promoter, followed by 2 open reading frames. The first open reading frame encodes for a ribonucleoprotein that basically shuttles genetic material to and from the nucleus. And then you have a second open reading frame that encodes for 2 important activities: endonuclease, the ability to cleave DNA, and reverse transcriptase, which is the enzyme that basically is utilized to carry out reverse transcription to make a cDNA copy of the messenger RNA, which is generated as a function of transcriptional reactivation of the element, to then shuttle back this cDNA into the nucleus, use it for target prime reverse transcription, and then make a copy that's eventually reinserted back in the genome. And hopefully what you can sort of imagine as a function of this description is that if in fact benzoate pyrene in a sort of reliable way reactivates these sequences, you now have generated a powerful means by which benzoate pyrene can amplify its ability to cause genetic injury through reinsertions back into the genome, which happen to be, random, or so most of us used to believe, and change programs of genes because the reactivation process is an epigenetic process, as I will show you in a few minutes, and that changes chromatin landscapes. I think something that's really important for you to remember is that we all carry about 100 copies of this retroelement, the line 1 retroelement, all of which are potentially activated, 5 of which are very, very active, and we study one of those. And the one that we study is the one that comes from retinitis pigmentosa, and that's all I will say related to that. For now, I'll take questions related to that. And so what this means is that then, because after 5 years I was convinced that in fact it was real, this was a true response to benzoate pyrene, we began a molecular journey trying to characterize the biology of the system. And this captures about 10 years worth of research, so I guess when you have 20 minutes, that's what you have to do. So we basically dissected out that 5' untranslated region of the line 1 element to figure out which responsive elements there might actually could account for some of that biological response. Here's a cartoon for both the human and the mouse with some differences and then some overlap. The work that we had done in the mouse had actually characterized an electrophile response element, an ARE, that it's the binding site for nerve 2 and is a site also recognized by members of the AP1 family of transcription factors that actually affords redox sensitivity to the reactivation of the element. And in fact, we sort of worked our way and characterized fully what the importance of those elements were within that promoter. Turns out that there's multiple ARE binding sites in that regulatory region. If you ablate those sites, you actually ablate the responsiveness to the carcinogen as well as to oxidative stress, emphasizing the importance of oxidative mechanisms in the reactivation of the sequence. In the human, although it is responsive to oxidative stress, the real major driver to reactivation appears to be an E2F binding site, which is located about, plus 422 within this internal promoter of the line 1 region. The binding site is actually a binding site that is active, and if you ablate binding of E2F to that site, you actually impede the ability of the element to be reactivated. Now, why is that important? It becomes really important because there is extensive methylation of the promoter region of line 1 element. In fact, the majority of L1 methylation in the human genome resides within repetitive sequences, and it resides within L1 elements, to the point that if you want to do global methylation studies, and you happen to be interested in methylation, in fact, you could use line 1 methylation status as a readout of genome-wide methylation. That's how much methylation you've got in there. But what it turns out, and work that we carry out in the lab, revealed, is that this E2F binding site is critical to the repressive state of the L1 element, and that the ability of benzoate pyrene and TCDD, by the way, Tim, to reactivate line 1 elements involves disruption in the silencing machinery that sits on this regulatory region. Because it turns out that this E2F binding site recruits retinoblastoma. And retinoblastoma, in turn, assembles a repressive complex that gives rise to silencing of the element. And I'll walk you through some of those findings in a few minutes. So, in order for us then to study the biological consequences of line 1 reactivation in this system, we had to engineer a way to be able to look at the biology of this system. When you've got 42% repetitive sequence in the human genome, 17 of which is this sequence, of course that means that your background signal is going to be a little high. And so what we did was we created synthetic versions of the line 1 retro element in which we encoded proteins which were foreign to the cell, the human cell, and then we could essentially use these tags, if you would, to follow the biology of the system. We also engineered into these constructs a reversed truncated antibiotic resistance cassette that would enable us to give us information about retrotransposition events. Because if when you read in forward orientation that messenger RNA has a neomycin gene that is disrupted, when the gene actually engages in reverse transcription, then the copy that's generated following alternative splicing is going to generate a fully functional neomycin gene, resistance gene, that could be used for selection. And so we now have in our hands a system where we could not only read out the production of protein, but we could also read out retrotransposition. We convinced ourselves that in fact this is important, and we applied this to understanding of, in this particular case, liver oncogenesis and whatever role line 1 elements might have in that. Suffice it to say for now that viral carcinogenesis, which of course in the context of liver is probably the most important aspect to human biology, Hep C virus oncogenesis actually involves reactivation of line 1 retroelements. And so these line 1 retroelements, I think, are going to be very important in our ability to understand the biology of cancer. We chose to use a Hep G2 cell model because this, of course, is a non-tumorigenic, transformed hepatic cell line that carries out a lot of differentiated functions in liver. So it gives us an intermediate state to understand what role, if anything, line 1s might have in progression of the oncogenic signaling cascade. Tim, you asked a question that I think has been repeatedly asked by many people, which is sort of what then is the connection between fibrosis and the occurrence of, cancer of the liver. And I think one of the critical answers to that question is that the occurrence of the fibrotic phenotype in liver in many cases involves transition of cells from a differentiated state to an undifferentiated state that actually affords that undifferentiated cell phenotype the capacity not only to regenerate uncontrollably, of course, in the case of cancer, but also to deposit matrix and to create a microenvironment that's going to be suitable for the cell to support itself. And one of the things that we found is that if you overexpress line 1, or if you challenge cells with benzoate pyrene, in this case, the HepG2 cell, but this applies to pretty much every single other organ system that we have studied, what you find is that the cells undergo epithelial to mesenchymal transition. And this is just a partial compilation of some of the genes that we've studied at the immunofluorescence level to convince ourselves that in fact these proteins were being made. And so what the story appears to be unfolding like is that BAP-related molecules, through mechanisms that involved epigenetic reactivation of line 1 retroelements within the human genome, give rise to transition of cells from a differentiated state, in this case an epithelial phenotype, into a mesenchymal phenotype that is actually capable of moving out, migrating, metastasis, and creating a microenvironment that's going to be suitable to altered biology. And so in trying to understand this phenotypic transition, and you can sort of reflect back to the first slide that I showed you, the plasticity of the genome and its ability to move out, in and out of these domains of genetic control, we actually turn back to something that we had done before, which related to a discretization of a line 1 regulatory network. Back when we did this work, this was really challenging because most of the tools that were in hand to be able to study genetic regulatory circuits were based on biological knowledge. And for line 1, there wasn't much of that. So I'll tell you details of that if you're interested later on, but suffice it to say that we have identified a 34-gene regulatory network for line 1 that involves 3 unique biological pathways that will resonate, I think, with many of you in the audience. Turns out that the regulatory control for line 1 involves disruption of genes in the inflammation cascade. Not surprising. It involves alteration in extracellular matrix genes, and it involves alterations in cellular metabolism. And if you reflect back on the talks that we have heard here over the course of a day and a half, these are repeated pathways that keep coming back every time that you think of toxic injury. And so what we found in the context of the EMT response in liver is that we had 92% fidelity of this regulatory network, and that the alterations, to our big surprise, did not always correlate with reverse transcriptase activity. And what that meant is that perhaps not all of the genetic alterations associated with line 1 reactivation as a function of carcinogen exposure required that complete cycle of retrotransposition, which I alluded to in the beginning of the talk. So reinsertion may not actually be completely important. Using fluorescence in situ hybridization, we actually studied 5 of those genes which were within the network, and what we found was that only 2 of those targets, only 5 of them though, but only 2 of those targets actually involved altered functionality of the target as a function of a line 1 reinsertion. And those 2 were VCAM1, very important in the context of hepatocarcinogenesis, because it turns out that when hepatic cells acquire a transformed malignant phenotype, they actually overexpress VCAM1, which is the matrix protein that changes the integrin regulatory profile of those cells, as well as a protein phosphatase that we actually have not studied very much. The other targets did not involve reinsertion, and of course that left us with the idea of how is it then that the genetic network is being altered, and more importantly, how is it then that we can understand the pro-carcinogenic activity of line 1 reactivation? And the answer to that is captured in this slide. Turns out that, if you remember, I alluded to the fact that the E2F binding site within the regulatory region of the line 1 retroelement was involved in the recruitment of retinoblastoma for formation of a repressor complex that epigenetically kept the line 1 retroelement in check. And of course, the real question for us became, what then are the accessory factors which are recruited as a function of the E2F-RB multimeric complex sitting on the regulatory region of the line 1 retroelement that was responsible for silencing? Unpublished work has actually, in fact, the paper is getting ready to go out, work that was done, here by Diego Montoya. Turns out that the NERD repressor complex is what drives silencing control of line 1 retroelements. And so when the NERD repressor complex is sitting on this template, you have a heterochromatin state that does not allow for either expression of the retroelement itself, and perhaps more importantly, chromatin states of the neighboring chromatin that you've got in there. When you've got reactivation of this element as a function of carcinogen exposure, there is removal of this repressor complex from the template, and in so doing there is unwinding of the chromatin, because this is what's going to allow the element to be expressed. But because you have such a vast prevalence of line 1 retroelements throughout the genome, you now have changed the entire chromatin landscape of that particular site. When you change that chromatin landscape, you now have allowed the cell the capability to reprogram its genetic profile. And so this then goes back to what we were talking about early on about the plasticity of the genome and the ability of cells to transition from one state to the other. So lines actually might be what empower cells in response to, in this particular case, chemical injury, but this applies to viral injury, it applies to stress injury in other forms. It allows, these lines might actually be engineered in the genome to actually allow cells the ability to transition from one state to another state in ways that do not necessarily involve mutational alteration. Because if a complete cycle of retrotransposition is not taking place, by simply opening up the template, you now allow for the complexity of changes that Tim showed you when he captured the genomic landscape of TCDD challenge cells. So I think from a toxicological standpoint, this whole idea that lines are targets of toxic injury becomes really, really important, because you can actually redefine the way that we think of, carcinogenesis, of chemical carcinogenesis, because now we have at least one way in which we can explain the intersection between what we understand on the basis of genetics. Now, Jim, much of like the work that you've spent, many years, understanding, the intersection of that with epigenetic control in cells, and maybe the whole sort of conundrum that we've always faced, trying to understand the complexity of non-genotoxic carcinogenesis. Of course, the question remains, how does this apply to many other molecules? We've only studied polycyclic aromatic hydrocarbons and metals, which also go through this pathway. I think another important message from here was, at least for me, the powerful biology that can actually be predicted in the absence of actually doing an experiment. That genetic network that I showed you was constructed fully in the absence of any biological knowledge, and I'll tell you the details of that if you're interested. I think a third important message that I want to leave you with is this whole idea of clusters of epigenetic control with lines actually serving this particular function. And then last but not least, the idea that if you then target the molecules which are recruited to either the repressor complex or to the activator complex that sits on these particular elements, you might actually now have a means of actually modifying the course, the trajectory of disease progression. And we actually are utilizing epigenetic therapies to try to downregulate the overexpression of the retroelement and see what the impact of that is in the context of phenotypic control of the cells. I'd like to acknowledge funding from AstraZeneca and National Institutes of Health for this work. In fact, the work that we did looking at the genetic network was done in collaboration with Ruth Roberts at the AstraZeneca. And then the people that actually did all of the work that I showed you, Pasano Bojan did beautiful work for the fluorescence in situ hybridization. I've already alluded to Diego's work looking at the repressor complex recruited into line 1. Alexis Hand, Marco Tavera, Ivo Tenning, a former student in the lab, Velas, Ty Heberly, and Irma that actually helped construct some of those constructs that I showed you before. I hope I have time to answer a few questions, and look forward to your reaction to what I just said. Thank you. Yes, Angie.
Kind of a general question, but so what's the relationship? Is there any relationship between line 1 and, say, an AFLD, or say just in general metabolism, and how metabolism may intersect with regulation of line 1? Does anyone?
So the connection there is that many of the genes that change as a function of line 1 reactivation are genes involved in regulation of metabolic control in cells. So that the metabolic deficits or the metabolic changes that you see during that transition from epithelial to mesenchymal might actually be driven through rearrangements, epigenetic rearrangements orchestrated by line 1 retroelement. We've never looked at sort of the functionality of that side because I'm mostly interested in the genetic piece, but the prediction is that those deficits are actually accounted for. The reason that you know there is a connection is that if you take a silencing RNA to line 1 and you actually halt the progression of the oncogenic phenotype, you prevent the metabolic alterations predicted by the genetic network. Yes, Gavin.
Very compelling seminar, and I don't have a question per se, but a comment. One of the things that we should keep in mind too is one of the salient features of cirrhosis also are these regenerative nodules.
Yes.
And these alterations may well be in place in that. That should be looked at.
Absolutely. A paper came out in Cell last year, actually tackling a little bit of what you're making reference to, looking at Hep C virus cancers at different stages, and it appears that the more advanced the stage, the more these line elements appear to be active. Initially, very sort of, I guess, implicitly speaking, we were thinking of this as drivers of genomic instability in cells, because obviously if you reactivate these things, now you've got chaotic, genetic rearrangements taking place uncontrollably. So very important point, yes, by all means. All right, yes, Craig?
You looked at hedgehog signaling at all?
We haven't, but obviously a very important area to look into in the future, absolutely. Yes, Gary?
Yeah, well, there may be a question in here, we'll see. But we had a talk last night after dinner that emphasized certain aspects of liver pathology during hepatocarcinogenesis, particularly the NTP bioassays.
Uh-huh.
And I just want to call attention to the fact that there's been a major controversy in the pathogenesis of experimental liver carcinogenesis for decades now, with one set of investigators emphasizing the emergence of what are called oval cells from the periportal region.
Yeah.
And these are postulated to be the progenitors of liver neoplasm. On the other hand, there's a group, myself included, that has focused on the transition of hepatocytes into neoplasm. Now, one of the lesions that Dr. Malarkey called attention to last night in their study was this cholangiofibrosis,
Yeah.
which is associated with oval cell proliferation. And I would submit that there's been a lot of molecular analysis surrounding that kind of scenario in the liver, which is probably irrelevant to carcinogenesis at more moderate doses of carcinogen. And from what I'm understanding from what you're saying here is your data would provide an explanation for how the hepatocyte could reprogram itself to emerge as a neoplastic cell.
That's correct.
And that would fit with the fact that with immunohistochemical markers, we can identify little clusters of 1 or 2 cells of hepatocytes early in the process of liver carcinogenesis. That's
actually, and actually, something else to keep in mind. I think that you're sort of capture what I try to convey for you. That is what I believe, that these cells have the ability, the plasticity, if you would, to remodel themselves into an oncogenic phenotype. The question is, where are they along this path, and when do you reach point of no return, right? What is the point at which you can no longer sort of come back to the normal phenotype? If you capture them before, you can actually potentially address, through intervention, the carcinogenic process. The other thing to remember...
Let me just quickly throw something. We've done a number of stomp experiments where we've quantified these lesions in
Yes.
different stages, taken the animals off the carcinogen for 6 months, the lesions go away.
Yeah, but I'm saying there's going to be a point that lesion's not going to regress. So, and the question is finding, what that point is. The other observation before I, return back to the podium is that the comments that were made regarding this sort of sensitized cells that might actually be the ones that give rise to oncogenesis relative to a parenchymal cell, fully differentiated, capable of then undergoing the differentiation, are not really mutually exclusive. They might just be cells which are sort of residing at different stages of this phenotypic spectrum, where you've got populations of cells within any organ that actually are primed for genetic reprogramming, and cells which would be more resistant to this genetic reprogramming, and that would require challenges like the one that I showed to be able to undergo that change.